Hi everyone, my name is Kimberly Mackay. I'm from the University of Saskatchewan and I'm here to talk about our poster called StoHi-C, using t-distributed stochastic neighbor embedding to predict 3D genome structure from Hi-C data. So just before we get started, there is the contact information for both me and my co-author Anthony Kuslik on the top of each and every slide. There's also a link to our preprint, so all of this information has been published as a preprint and hopefully will be published in a journal shortly. So if you want any more information, you can always take a look at that as well to get a better idea of what we were doing here. And additionally, all of the code, all of the results, all of the input data sets to generate these results are all available on my GitHub page at this link right here. So if you want to, you can go ahead for yourself and give this code a try. If you have any problems, always just post on the GitHub page and I'll get back to you there about how we can try to fix those things. So instead of going through my PDF of my poster, I thought it would be a little bit easier in this format to go through a set of slides instead. All of this information is also included on the poster PDF, and it is just a little bit more of an easier way for me to kind of get the message across in this virtual format. So hopefully you'll find this complimentary and not confusing. But the problem that we are working with is something called the 3D genome reconstruction problem. So I'm going to just assume at a very high level that everyone tuning in knows what Hi-C data is. If you have any questions about it though, please just reach out to me. I'd be happy to answer. But just for the sake of time, I'll assume that everyone's familiar with Hi-C data, and the process of running a Hi-C assay generally produces these things called interaction frequencies. And so what they are is a count of how often one region of the genome was found in close proximity with another region of a genome and then normalized to remove all these biases that we know are inherent with Hi-C data. Often we're going to visualize that as something called a whole genome contact map. So you can think about the linear extent of the genome being stretched along the X and Y axis, and then those counts just being represented with a color within this matrix here. So the stronger the color, the higher the count. What we do with the 3D GRP is we take those counts and we actually convert them into XYZ coordinates for each region of the genome, and I'll often call that a genomic bin. And so by doing this, we're able to get a prediction of the structure of the 3D genome. So similar idea to what we would see with like a protein structure, but now we're looking at genome structure. So there's lots of different methods that have been generated to solve the 3D GRP. Many of them use multidimensional scaling to do this. And when you look at the problem and strip away all the biology, MDS, or multidimensional scaling, seems like a really good solution to the problem. But when you actually apply it to Hi-C data and you're applying it to the biology that's inherent to Hi-C data, there's lots of problems that come up that show that maybe MDS isn't the best method. Now, a lot of the existing methods have been able to overcome a lot of these problems by doing things like very creative divide and conquer approaches so that they can still use MDS and still get biological results at the end. But I was wondering if we could come up with a better solution to remove some of these more computationally expensive divide and conquer techniques and just rely on, say, a different kind of embedding technique other than just MDS. So one thing that hasn't been used very frequently within the community is t-distributed stochastic neighbor embedding, or t-SNE. So it's very popular in other facets of bioinformatics and other facets of, say, data visualization and that, but it hasn't been applied to the 3D GRP. So we developed this new workflow called StoHi-C, and all it does is it takes our whole genome contact map as input, uses t-SNE to perform the three-dimensional embedding, and then we take the XYZ coordinates generated by this first step here and input them into a visualization tool. So on the GitHub page, there's 3 different options, but really you can use whatever kind of visualization tool you would like as long as it takes XYZ coordinates. So we have specifically coded up Matplotlib, ChartStudio, and Plotly. So a lot of my biological collaborators really like ChartStudio because it's a lot more interactive. You can zoom in and move the graph however you like. The Matplotlib output was going to look something like this over here. So it's just a simple animation with a rotation around the y-axis. And then the Plotly is going to be very similar to the Chart Studio one. So that's just an overview of the workflow. It's quite simple, and we wanted to test it out on a data set to see if it was going to work reasonably well. And so the data set we chose was a fission yeast data set. So we have 4 different types of yeast. We have a wild type, a G1 arrested, RAD21 mutation, and CLF4 deletion. What's really interesting is that these data sets, these StoHi-C data sets, have been around for a long time, but none of the existing tools have been able to successfully predict 3D genome structure from them. The existing tools are very good at predicting 3D genome structure from things like human data or mice data, but this fission yeast data was still a bit elusive. So what we did is we ran the Stoic workflow on all of these data sets. You can see panels A, C, E, and G all have that Stoic workflow. And then we also ran this data set with an existing MDS method, and those results are presented in panels B, D, F, and H. And so you'll see, if you're familiar at all with known hallmarks of fission yeast genome organization from the years of microscopy images that already exist in the literature, that with the Stoic workflow, we're able to get a lot more biologically relevant structures appearing in our predictions. So things like the RAB1 configuration of the chromosomes, where centromeres are clustered on one end and then telomeres are clustered on the other end, and the chromosomes form this little hemispherical region. So that's a brief overview of the results. In the future, we're looking at applying this to more complicated genomes, larger genomes, to see how well it scales. Also looking at other kinds of algorithms, so different ways of implementing t-SNE, looking at different algorithms that may be a little bit better than t-SNE to see if we can do any better than this already, but it seems to be doing a lot better than the MDS methods. Thank you.